Great. And so we'll start screen sharing when we're ready as well.
Ready to start? Good to go?
Yep, so should be good.
All right. So welcome everybody. We're on Zoom today for our partnering seminar. And today it's a great pleasure to introduce firstly Allison Pettit from the Mater, who's had a long interest in osteology and bone immunology and regrowth and bone defects.
So
We've got a problem?
I can just move the share screen so it'll be a bit cleaner for you. So, there you go. And her translational partner is Professor Martin Wullschleger from who's the Director of Trauma at Metro North. So first Alison will speak and then Martin. So welcome.
Thanks, Ranjini. It's really a pleasure to speak to you today, and especially to be here with my clinical collaborator, Martin Wullschleger, who we've worked with now for over 10 years, and he's really made a big impact on the translatability of our research. This story started when I came back from a postdoc in Boston and joined Professor David Hume's laboratory. And David has a career-long obsession with tissue-resident macrophages. And so when I joined his lab, it was only appropriate to start to develop some research projects that were aligned with his passion. And as most of you will know, tissue-resident macrophages contribute to organ homeostasis, and they adapt tissue-specific phenotypes to the tissue and even sometimes the microenvironment within the tissue that they exist. And these macrophages are mostly dependent on the growth factor, colony-stimulating factor 1. And when I first joined the lab, David happened to mention to me that in one of the studies he'd done in the mid-80s, he'd observed that there were FR80 positive macrophages present within the tissues lining the bone surface. And so it's not a very straight surface here, but that's the bone there. And I might even try and put the pointer on. Bone surface there, and here in the tissue, aligning that, you can see these. I
think it's just not
screen sharing.
Back to this share screen. Sorry. Let's try it again.
Places you could be.
For sure.
Sorry, I'm just going to try and.
Come give you some more support. Yeah, it's coming too.
I'll go with this. Let's see. Okay.
Yes, sorry. So sorry about that. We just had the technical issue of sharing the screen. So backtracking just a tiny bit, when I joined David's lab, he mentioned to me that there are FR80 positive macrophages in the tissues present on bone surfaces. And in this image that was published in the mid-80s, you can see here there's the bone surfaces lining here with the matrix at the top of the image, and within that tissue are FR80 positive ramified macrophages. And so this had not been really noted in the bone and mineral field. And so in collaboration with another postdoc who joined the group, Dr. Liza Raggett, we asked the question is, do bone tissues contain a tissue-resident macrophage population that has tissue-specific function? And through a large amount of research involving students who joined the group, including Ming Chang, who is the first author on the paper we first published in 2008, we found that there are indeed FR80 positive macrophages present on resting bone surfaces on the inside or the endosteal surface of bone, as well as the outside lining tissue or the periosteal surface. And when we looked more closely at areas areas of actual dynamic bone change, we could also find these macrophage populations associated with bone formation, bone turnover or bone removal by osteoclasts, which are the big red cells, and the F80 positive cells are adjacent to those bone resorbing cells, as well as within the complex but basic multicellular units which coordinate bone resorption at the leading edge and bone formation at the tail, which include F80 positive macrophages in the canopy that is containing these bone multicellular units. So we presumed, based on this very stunning distribution, that they would have tissue-specific functions. And we called these resident tissue osteomacrophages, or Osteomacs for short. And so the approach we took was to use an inducible macrophage depletion approach. And here in our macrophage replete samples, at the top, you can see the F80 positive macrophages are sitting on top of these big plump cells that are right on top of this bone surface, and staining in serial sections showed that these cells are the bone-forming osteoblast cells. But when we deplete macrophages, we don't just get rid of the macrophages, we also deplete the osteoblasts. And through a series of experiments, we showed that the loss of osteoblasts is an indirect effect of the macrophage depletion. And then using in vitro strategies, we showed that for osteoblasts to mineralise matrix in response to the physiologic stimulus of elevated calcium, you required macrophages to be in with that co-culture, whether they were macrophages isolated from bone tissue preparations or in vitro generated bone marrow macrophages. So we provided the first evidence that osteomacrophages that exist in bone lining tissues are able to support the bone formation function of osteoblasts, demonstrating a tissue-specific function for these cells. So that was a very nice knowledge gain for the field, but what were the potential health applications of this? And the very common disease osteoporosis was something that immediately seemed to be a logical application of this knowledge. Osteoporosis causes bone fragility, and it's silent in that you don't actually appreciate that you have osteoporosis until you present usually with a fragility fracture. And these fragility fractures commonly occur in the spine, hip and forearm, but also in other areas. And they start presenting fairly commonly from the decades of 50s and with increasing incidence as you get older, and particularly in women through the loss of estrogen after menopause. They're extremely common and they're incredibly costly. And more importantly, fragility fractures actually double the mortality rate and triple the mortality rate for women and men respectively. And this extends in the 5 years post the initial fracture period. So these fractures have a significant impact on the longevity, and as well as create a large amount of morbidity for the patients who suffer from them. And what we were particularly interested in is that after a fragility fracture, 50% of the fractures show delayed healing. And so this actually extends the period of morbidity associated with a fragility fracture and has a large impact on the patient's quality of life and recovery, that there's some indication contributes to the increased mortality. The other potential application is this sort of scenario of extremely complex trauma-induced fracture, and these fractures are starting to also become more common based on World Health Organization studies and are incredibly challenging to treat. So we identified the treatment gap being that there's no broadly applicable routine frontline therapy for fracture. So the predominant treatment approach is orthopedic management, either through stabilisation and immobilisation, or through surgical approaches, which are expensive and requiring hospital stays. There is one single therapy that is available for use in open tibial fractures, but this this therapy has a high risk of inducing cancer, so it is not ever extended to broad applicability across other fractures. So we feel that there's an unmet need of developing therapies that can accelerate fracture repair in healthy bone, because even a normal fracture has an extended period of morbidity and with financial implications. We feel that there's an opportunity to prevent or correct delayed fracture repair healing in high-risk groups such as osteoporosis. And there's also delayed healing in diabetic individuals, so another large population of potential application, and also to complement orthopaedic strategies in complex fractures. And so when we were deciding how to apply the knowledge gain, we first went for a very simple model that we could easily implement in our research team, which basically just involved simply drilling a small hole in the distal tibial crest, which then goes through a fairly predictable healing response. But it only heals by one of the types of potential bone healing mechanisms, which is intramembranous ossification. And we were very easily able to show that depletion of macrophages in this model severely impaired the healing response, suggesting that macrophages are very important in this process. So while this was a very useful experimental model, it doesn't have much clinical relevance to fracture healing, because it's not often that you're just healing a simple hole in your bone. So we were looking around for what fracture models we could use at the time that we were wanting to do these experiments. There weren't a lot of good options in the literature. So there was closed unstabilised fractures, where you can see here that there's really poor alignment and reduction of the fractures, creating a huge amount of variability in the healing response, and not to mention the ethical concerns. So the clinical relevance of this is also low. Or there were models that tried to replicate intramedullary nailing of a fracture. And basically these would just use a needle, a hypodermic needle that was chopped off and pushed into the bone. It wasn't fixed into place, again creating a lot of variability. The needle itself is not well designed, with bending of it a common occurrence, or it would even be left with large protrusions outside each end of the bone. So we didn't feel that these were appropriate models to progress with. And that's when I attended a conference that I wouldn't normally go to. It was the Australian and New Zealand Orthopaedic Research Society conference. I was invited to present at the conference, which was incredibly lucky because Martin was also presenting at this conference. And that's where I found, a really useful clinical partner. And so I'm going to hand over to Martin now.
Thank you, Alison. That was in Canberra, or where was that? I think it was.
I think it was.
Yeah. Alison, thank you very much for inviting me. It's a pleasure to be here and just to share our experience, I guess. When you started off with saying that periosteal macrophages are important, then I sort of nearly stepped in and said, well, I knew that before. It's interesting that from the clinical side, one of the first things I remember that was hammered into my brain and my practice as an orthopedic trauma surgeon was to look after the periosteum. And yes, orthopedic surgeons, at least some of them, it's more about bone and joints and all that, and not so much about soft tissues. But we particularly around the AO, where we got trained, my training was in Switzerland. It was very pedantic, particular to really just take a millimeter, maximum 2 off the fracture side and just leave the rest intact. And yes, we did have really good outcomes with that. So that's sort of from the clinical side, which fits very well with what we're talking about. The other thing is coming from Europe here, finding my feet I did a PhD with QUT in fracture healing, a large animal model, in fact, but I was a bit bored. Also as a surgeon, not doing too many surgeries, I was sort of looking out for areas where I can help other research groups. And yeah, there we go. Then we met, Alison and I, and just brainstormed on where we could actually take the clinical and the research to a basic research approach together. And as orthopedic trauma surgeons are, we like instruments and technical things, I guess, and to play with stuff, and that was really a great option. And I'll just show you a few pictures later. So basically, what our model needed to include is that we have really reproducible fracture. That's one of the first things I learned in research, fracture research. It's just the reproducibility. We can't study it if it's not reproducible. And I guess in fractures, that's a hard thing. Knowing from the clinic again, every fracture is slightly different, and that's for us. It's challenging. I guess that's a nice thing, that it's not getting too quickly too boring. But for research, it's very important to have that reproducibility. And then how to create a fracture. I mean, ideally, we would have a fracture created, again, very reproducible in terms of the impact, but then also in terms of the pattern and the reduction or the displacement. And that can be done with some blunt trauma. So there are drop methods on legs. But as you can imagine, sometimes that doesn't go the way and it splinters everywhere. So it can be quite tricky to actually control that. The 3-point bending in theory is great, but particularly talking mice, that's then really tiny, and to get it right in the right spot that you afterwards being able also to fix it. And then that's the next problem, because mice are so tiny. Once the fracture is displaced, then to actually reduce it, that can be a challenge. And if you're not as experienced in fracture fixation, it's nearly impossible. So then we are sort of going towards osteotomy. Now, osteotomy, for sure, it's not, I guess, a true fracture. And we have some damage that is different to fracture lines on the surface. Because if you drill or with an oscillating saw, you create some osteonecrosis. If you do it gentle and careful, you might not have that much, but you still have a bit damage. So in those terms, an osteotomy is obviously a bit different, and also it's quite one direction, which again is more reproducible, but on the other hand, it doesn't really reflect a normal fracture pattern. But so we had to sort of find the way in between. And then in terms of reduction in mobilization, we really wanted a method that we can, I come back always to the reproducibility, but well, that's basically where it comes down to. So that you have an implant that you know exactly the biomechanics. And at the time, in the clinical environment, we went more and more towards implants that if you put a plate on with screws, that the screws actually hold in the plate, so locking screws. That was quite new. That started around 2000. And so to make that actually then also happen in mice, which again is tiny, that was a real benefit. And they actually, well, it's a friend of mine that developed it in Switzerland. And so I like the tool anyway. So that's what we then used. And it was good, and you see here the paper we did previous just to basically validate the model and the implants. We fixed plates on those bones and then studied with different implant options, and I'll show it to you here. So we have, the first was just a normal plate. You see here, 4-hole plate. And then the other one is a 4-hole plate as well, but had actually on the sides here has small wires. So you can imagine that it's a bit flexible, that implant. So we had now 2 implants, that's to start with, and we had different wires also for the sides of the more flexible plate that we studied. And you just evaluated and validated that model. And the study that we quoted before is really that, as expected, with a more flexible plate, you have more callus, and it takes longer to get to your rigid and torsional stiffness in the mechanical test. We actually did test those very tiny bones once we explanted them in Mous up at the north side, the Prince Charles Hospital lab. And yes, but interestingly, after 21 days, it caught up. So the colors in the flexible plate actually caught up nicely and was actually basically as strong as the other plate. So it was a really good model to study. Now, a few details is, so you have a cheek that you put the screws in. You see it here with the mid-shaft fracture or an osteotomy, which we created after we put the plate on. And the screws, basically, they're on top of a pin, and you just drill them in, and then they break off. And it locks here in the plate. So very reproducible, fancy, I guess. It's not too cheap, unfortunately. But well, that's why we have grants for. Okay. So here then, you see a couple of clinical pictures, and it was great to study really over time. So first we set up the model to have, as you should, to have really good baseline results just without anything, just the implant basically on. And you see here, we did it mainly with the wire plates because we saw more colors there and we really wanted to study that a bit of delayed colors formation. And you see here very nicely the histology of the inflammation first, after 7 days, 14 days, the soft colors, and then more the hard colors here, and the really nice hard colors actually, in fact, and you see here opposite the plate. And then we proceeded, we wanted to get really fancy and to get to, because that's one of the problems clinically as well, that you have large gaps. And yes, we do actually in MERF as well, large defect studies in sheep mainly. But for certain things, obviously, we know that small animals are better to study more the molecular as well as the cellular basis. It's far easier. And as we had the established group, so we just went into this gap healing as well. And this one here, that was just was that one? No.
That one had no implant.
Yeah, so that was a control group, and that did not heal. It just did not heal. After, I think, four weeks. 4 weeks. Some even left a bit longer, but 4 weeks, there was no colors whatsoever, so that wouldn't heal. And then here we put it in and scaffold.
Biomaterials, bond scaffold.
Yes.
Size.
And it healed beautifully. It actually had colors and did we do mechanical tests?
No,
really.
No, we haven't progressed that far yet.
But so that definitely healed well, and wasn't any complication, no problem. And then just to simplify it a bit, because the plate model, as I said, it's quite expensive, and we just wanted more a simple model as well as an intramedullary. So in the clinical environment, it's sort of a bit well, an argument, I guess, to a certain extent, whether you go intramedullary or to fix, or you go extramedullary with plates. And for certain fractures, it's clear. If it's close to the joint, nailing is difficult and normally not successful. As mid-shaft, you should basically always use a nail, and it just heals better. And because we do mainly mid-shaft fracture models, we thought we'd go back to more the intramedullary. And then Romano, my friend from Switzerland, developed this new Mous screw, which we then started to use in the femur retrograde insertion. So from the bent knee, so you bend the knee and you drill a small hole and then you pin it up. And then once it's in there, you create the fracture with a small weight that comes across. If you do the weight first, if you break it first, then you struggle to reduce it again and actually pin it in the right way because it's so small. So this model is, apart from being cheaper, it really, as you see here, it has has low failure rates and it's really reproducible, particularly around the fracture side. So it's a good model as well. Okay, I'll hand over to you again.
Yep. Thank you. So with Martin's assistance, he would regularly make time in his clinical schedule to actually come in and assist with these surgeries.
So we do the surgeries after hours?
Yeah, to make it work.
So I stressed a bit on the team, and have to acknowledge you guys. I know that you don't work after hours so much, but we do. And so we had a lot of, not necessarily night shifts, but evening shifts.
Yep.
So there was definitely nobody else than us in the lab, but we had lots of fun and good outcomes.
Yeah, and the animal facilities that we've done these in over the years have also all been really helpful in actually enabling us to do that based on the schedule. So we should thank the BRS as well. But using this, the much more clinically relevant models, we were able to reproduce our findings that we came across earlier, and using macrophage depletion approaches, we're able to demonstrate that macrophages were critical for achieving optimal healing, as you can see quantified on the edge here. But then we were also able to use interventions to see if we could actually improve the healing outcomes. And this shows us using the macrophage growth factor CSF1 as a therapy, and we were able to enhance the fracture outcomes through CSF1. But CSF1 has a very short half-life, and it's not a particularly good drug. And so again, we decided we needed to improve the clinical relevance of this, and I forgot to put David's picture on the slide here again, but David's research team had actually developed a chimeric CSF1 molecule that's coupled to an FC, which greatly improved the drug qualities of CSF1, but there was a risk that this would actually cause catabolic bone reactions, and so we had to come up with a therapeutic strategy that did not lead to bone loss, but was sufficient to amplify the bone healing response. And this is a lot of work done by now Dr. Lena Batoon, who just had a PhD conferred. And so again, using this drug, using this drug-like molecule, we're able again to show that we could enhance fracture repair using the plated model, and this time use the more clinically relevant measure of fracture strength to demonstrate that it did improve fracture healing, and all the collaborators involved on the slide here. Now, this is important because these fractures really are healing pretty much to their maximal biological outcome because there is no stress on this. So the fact that we were able to enhance the healing in that situation, we were quite excited about. But again, our main purpose was around post-menopausal osteoporosis fragility fractures. And there are no really, and when we went looking in the literature, again, there were no really reliable or appropriate models of delayed fracture healing after OBX in a mouse. And even the rat models were all fairly not really reliable to use. And so using the intramedullary pin model that we had access through, again, through Martin's collaboration with Romano in Switzerland, we were actually able to develop a model of delayed fracture healing in mice using the pin model. And again, it comes down very much to the reproducibility of this model, the stability, the quality of the fixations by the fact that the pin had screw attachments at both ends compared to other models that really allowed us to do this. And you can see here the bone volume formed after OBX is significantly reduced, and the fracture non-union is significantly reduced in this model. And so based on the fact that we now had a reproducible model of fragility fracture, we then again applied our CSF1 FC fracture therapy, and we've just had this accepted for publication in Biomaterials. We're able to show that we could enhance the fracture healing even in the setting of postmenopausal osteoporosis that we're really quite excited about. But even with that, CSF1 has no protectable IP, and the amount of prior art indicates that simple mode of use pattern is unlikely to get through. So we still are at a bit of a roadblock with how to translate these observations. And so we're now starting to do work in collaboration with some pharmacists, ways to look at novel targeted delivery mechanisms, or coupling it novel ways to biomaterials to actually achieve this to be a therapy that we can apply to fracture healing. Our other alternative is to actually identify other novel molecules that are expressed by this macrophage population and translate those. But there's lots of things that happen along the way, and this discovery actually led to the generation of discovery of a molecule that has positive impacts on hematopoiesis that we are also currently trying to translate. So I think through our collaboration with Martin and being very much more focused on the clinical aspects of the research, we are really achieving more than we would have without that partnership. And that's just the funding partners, the funding support. So thank you very much.
We have some questions. Chat. Thanks.
You're welcome.
Pascal, pardon my naivety, but is rejection of the metal pins a challenge for treatment?
Martin, do you want to answer this one?
No.
Simple answer.
No. Our stainless steel and titanium implants, they are inert. Allergy is very, very rare, particularly with titanium. Stainless steel will be higher, but it's very, very minor. So no, we don't have a problem.
What about infection?
Infection, yes. So
although we have very low infection rates.
We in the lab, we did have.
Yeah, so the surgery is done, and particularly because Martin was performing the majority of the surgeries, it's done in pretty much clinical type scenario, and so we had very low infection rates, very low failure rates at all. The biggest problem we had with failure rates was with the large defect model, and the strains that are placed on the screws because of that larger defect are quite high, and C57 black cyst mice genetically have a fairly low bone mass, and so their bones were not strong enough, and we had to shift to different mouse strains for doing those. And we do get foreign body reaction to a lot of the implants that we're putting in, and so that's something that is a challenge for us to deal with. But infection itself is not really a problem.
And also we euthanize the mice early. The infection often comes a bit later as well. It's not something that comes straight away with a closed fracture. I mean, in clinical daily life with the compound injuries, open dirt in the air, that's a different story. But if it's in a septic and aseptic environment, you have a very low. Yes, that's right.
And we autoclave everything before it goes into the mouth, etc. So we've got a question, do we face any animal ethics issues? No, we didn't, because we particularly chose these models because of the fact that they were, compared to other fracture models out there, they were extremely good at reducing the impact on the mouse, and that you were retrieving stable fixation immediately post or even before the fracture was generated. The mice were provided with pain relief in the first few days post-surgery, but immediately normal ambulation usually occurred. So if a mouse had a limp, we assumed there was an issue with the fracture fixation. So the ethics committees actually appreciated the fact that we'd gone to a great deal of effort to apply a model that was much more ethically acceptable than those that were out there.
Yes.
In your experience, what would the usual lag time be between starting out with the initial animal model all the way through to getting sort of TGA approval, I guess, for a pharmaceutical product? Like, is it depressingly long?
It's depressingly long.
I guess it depends on the new technology or new method. It's up to 3-5 years, but in our case, it's much longer.
I mean, it's also pretty demoralising that we've shown that CSF1FC is effective, but we're not going to be able to translate that because of the fact that there'll be no company that wants to pick it up to further develop it, because they can't patent it and they can't protect the IP. And so that's something that happens quite regularly in research as well, is that the whole process of intellectual property protection is a barrier to translation.
Have you tried other strategies like chemokines to bring macrophages back?
So there's a whole other story around that we've been trying for years to so the question is whether or not we can use chemokines, etc., to bring macrophages into the site to replicate it that way. There's a whole other story alongside of integrity of data, and macrophages are actually more difficult to work with than you might anticipate. So we've tried to do this multiple ways over the years, but we've never been confident the data was actually correct. And we now have more recent information that suggests that that hesitation was appropriate. And we're now taking more strategies to make sure that when we do try to progress something like that, it really is the macrophages that it's influencing, or it is what is expressed by the macrophages. We don't think that we'll be able to really replicate what the macrophages do. So your suggestion of using chemokines to bring more of them in is probably the better way to go, because they do so much in the fracture site. So no single drug is going to replicate everything they do, and they change what they do depending on the time during fracture healing. So you need more macrophages there to respond appropriately to the signals that they're getting at the site.
Got another suggestion. Use bisphosphonates to couple whatever you want to do.
Yep, so we're considering that as well. So Ranjani has also suggested that we could use bisphosphonates to target the drug to the bone. So bisphosphonates are one of the most common treatments used for osteoporosis. They naturally target bone, and they have a toxic impact on osteoclasts that stop bone resorption and protect bone. But there are also bisphosphonates that don't kill the osteoclasts, so don't have an impact on the cells, but you can couple drugs to them, and that lead to targeting of a drug to the bone site. That is something we're considering, and whether or not that will be IP protectable is probably not really have a strong case either, but there are other companies out there that have got other ligands that do a similar approach that we're considering. I think we're probably over time, and there's no more questions online. Fantastic. Thank you, everybody.
Thank you.
We just end.
Yeah, just to end, and then